Hi everyone, and welcome to our next video on active transport. So this will be the second one out of 3 videos that we're going to have, and I'm just kind of ending off the screen with the last place that we had it located at, which was the sodium-potassium pump. So the first thing we're going to talk about is vesicles, which was a term that we covered last chapter. And if you remember, that vesicles last chapter was basically a membrane-bound sac that would be carrying materials that the cell may need, and it might be entering the cell or might be exiting. So one of the reasons why we might need a vesicle is because a lot of things are still too large to move across that cell membrane. For example, polysaccharides and proteins. And vesicles can help move those substances. And there are 3 types of motion that we'll get, or movement that we'll get, with using vesicles. And I got Annabelle here learning with me. You want to say hi, Annabelle? So she's going to learn about vesicles as well. So the very first one is endocytosis. Remember that the prefix endo means in. So this is vesicle movement into the cell. So this is movement into the cell by engulfing and surrounding a large substance. And there are a variety of different steps with this, and both types of endocytosis use these steps. So first of all, the substance approaches the cell membrane. The next step is that the membrane will form a pouch around the substance. The third step is the pouch pinches off and it separates from the cell membrane. And at that point, we get that vesicle. So we're using that lipid bilayer of the cell membrane, and we're just surrounding a material with that. And that's going to protect it from the cytoplasm, and it's going to allow it to move to where it needs to. And then this vesicle will move into the cytoplasm. So here's a picture I have of endocytosis. So the cell is the light blue, and the dark blue dot is a material that it's trying to swallow. And as you can see, the cell membrane is starting to kind of move around in the first picture of the object. Second picture, you can see the cell membrane has really moved around it. And the third picture is completely surrounded it, and we have kind of like a little bubble, a vesicle that's surrounding this object that we're trying to bring into the cell. So that's an example of endocytosis. Now, there are different types of endocytosis. The first one is phagocytosis. And it comes from the Latin word, which means to eat. And so this is movement of a solid material. An example I have right here is white blood cells. They will devour bacteria. It's kind of how they destroy them. So they'll take and they'll form a vesicle. Those vesicles will move to a lysosome and destroy the parts. So here we have a bacterium onto a cell. And that cell's actually moving the cytoplasm around the object. And eventually that object is now a vesicle inside of the cell itself. And we kind of talked about beforehand, that's how we thought that the mitochondria was formed as well. At one time it was something separate because it has its own form of DNA different than the nucleus. So it was probably like a bacteria cell or something like that that got swallowed. And then it started kind of functioning on its own and worked with the cell. All right, we're going to watch the videos when we get into class. But the next one is penocytosis. And penocytosis means to drink. So this is movement of liquid materials. So you can see in the picture here, I have a variety of different particles. Here, the yellow line is my membrane, and it's going to kind of pinch towards and then eventually form a vesicle, and that vesicle is going to move into the cell. The last one is receptor-mediated endocytosis, and this is forming vesicles on the membrane, and those vesicles have certain receptors on that membrane. So here is an example of receptor-mediated endocytosis. So I have this compound, it has certain molecule shape at the end as it comes to the cell membrane. There are actual receivers. They're going to receive this signal or this molecule. And then as it receives it, it's going to then form the vesicle, and then that vesicle is going to move into the cell. Okay, so this is as far as I want to get for today. I know I kind of flashed to the next slide. Next time we will be going over exocytosis. Can you say bye, Annabelle?
Bye.
All right. Hope you enjoyed this. I don't know, maybe if I can get it over with. All right, now say bye again.